Hi. So my previous video I have explained the cell cycle control. Now in this video you're going to learn the cell division mitosis. So mitosis is the pace you have seen after G2. So what is mitosis? Now mitosis is defined as a division of the nucleus. So nucleus division is very crucial. I highlighted many times the DNA is huge molecules. Why it is huge? It has 3 billion base pair, 3 billion base pair. So 3 billion base pair, they have a special sequence, special order. Now this sequence should be copied exactly to the next cells. So that's a crucial, the mitosis happening right way. So we have seen how it regulates. Now mitosis, again we're going to divide into several phases. So we have these, the phases come under PMAD. So what is PMAD? So these phases are prophase, metaphase, Anaphase and telophase. Now why we have divided into the 4 phases, it's easy to understand the process. So this process is just like a movie. So it's going from one end to another end. So we can't say exactly what's happening at one phase. But we summarize, let's say the whole process is like this. So we can again say beginning of the prophase, end of the prophase. So beginning of the metaphase, end of the metaphase. Beginning of the anaphase, end of the anaphase. Beginning of the telophase, end of the telophase. So in between we can discuss something. So actually what we are doing, just a summary of that, of all happening, the important events, we're going to show in this video. Otherwise, it's a continuous process. But we just want to take one snapshot and make a summary and show it to you how this is happening, what are the important steps. So, let's start with the prophase. So, I'm going to delete the slide here. So, start with the prophase. So, let's look at the appearance of the cell during the prophase. The cell membrane. That's a nuclear membrane. A prophase, the remarkable feature is we can see the chromosomes condense. So now chromosome condense, if someone wants to understand what is this chromosome condense, if we take a cell, the nucleus, the chromosomes are just like a cloudy. So just like a cloud, it's everywhere spread in the cells. Within this cloudy appearance, sometimes we can see a dark, just like the DNA, very dark areas and also very light areas. So how I can explain this one, DNA strand is like this and certain area it's very tightly packed like this. And a DNA strand again like this. So DNA and the proteins make this chromatin. This chromatin spread all over the nucleus in the light area and dark area. So light area and dark area, it's spread like a clouds all over. So we can't see a very clear appearance of chromosome. But prophase, we can see a clear appearance of chromosomes like this. This is due to condensation of the chromosomes. You can compare picture A and the picture B. So picture B is very clear inside, it's very condensed and X shape sometimes, the chromosome shapes are there. So this is the first picture you see in a prophase. Now, as I told you, just going to the next step, what happened? The nuclear envelope breaks down. So here the nuclear envelope breaks down. Breaks nuclear envelope. And you know, inside there is a nucleoli. So nucleoli start to disappear. It's also break down. So these events start during the prophase, as well as the centrioles. Centrioles already make the spindles and try to move to the poles, this pole and this pole. So at least we can see very few characteristics which belongs to prophase. So number one is condensation of the chromosome and visibility of the chromosomes. So disappearance of the nuclear envelope, disappearing the nucleoli, and centriole, spindle form, and migration to the poles. So these kind of 4 characteristics visible to identify the cell is in the prophase. Now once prophase complete, cell is moving to the next stage, metaphase. So let's look the metaphase. Now in metaphase we do not see the nucleus, the nuclear envelope. Instead we can see chromosomes like this. Now this chromosome, the middle region here, it's called chromatic chromosomes. I'm sorry, it's a centromere. So I will show it again here. That's a chromosome. Here it's a centromere. Now centromere, the function of holding newly formed the DNA. Let's say this one, A, is the original DNA strand. After replications, new DNA strand is here. So I just show it here. That's the DNA strand, and that's a next DNA. So that's the next DNA. This is DNA 1 molecule. That's the second molecule of DNA. So this is called sister chromatid. Sister chromatid. So this is the appearance of the chromosome now. Now here the centromere is attached to another structure called kinetochore to the spindle. This is spindle formed by the centriole, and this one is kinetochore. So I can show it here in the cell now, how they another one going to attach here like this, maybe another one going to attach here. So this is called metaphase plate or equator, because they align all chromosome in the middle of the cell's equator. And we can clearly see this kind of appearance during the metaphase. So if you look really the photomicrograph, we can see something like this. They are get together all like this appearance in a real cell. The cell is like this. So once metaphase is done, the end of the metaphase, the spindle starts to become shortened, and then they pull to this pole and this pole. So let's say what happened in anaphase. So anaphase, the centromere breaks and they separate the 2 molecules of DNA. So one is like this now, so one is like this. So they detach from the centromere and still attached to the spindle. And this goes here, this goes here, because the spindle becomes shorter and shorter now. As a result of that, we can see this kind of appearance in the cell. That's the anaphase. When the anaphase is completing, that means they are going more and more towards the poles, we comes to the telophase. Now in telophase, again the visible chromosome, the condensed chromosome, start to a little bit get loosened, and then we can't see real structure of a chromosome then. So they're going to not so visible here at this end, and maybe again the chromosomes are here, not that visible, because the prophase it's clearly visible, but telophase again they're going to a little bit relax, and we see this kind of appearance. And then the nuclear envelopes start to form. Also we can add here a nucleolus start to form. So in typical photomicrograph, we can see this kind of appearance of the cells, just like a two nucleus in a one cell. That way we can identify. So that's the end of the nuclear division. So this is the end of the mitosis. So now this stage, the nucleus divide into two, and we can see two nuclei in this cell. Now when this event is completed, I think we can add the last stage or phase, cytokinesis, because cytokinesis is a quite simple step. In cytokinesis, the two nuclei is here, and they're going to divide in the middle the cell. The cell membrane divide. So animal cell, this is called cleavage furrow, and the plant cell, this is called cell plate. So this division is important. The Golgi apparatus, they get together and they have a kind of, the sacs. These sacs try to be enlarged in the middle region, and they try to separate here, and finally we can see two cells, identical cells, at the end of the cytokinesis. So this is now originally the cell cycle control, and the mitosis. Everything now, these three videos, you will see how cell divide into two identical cells. If you like this video, please subscribe, and thank you very much.